This is a update of the webinar on cross-frequency coupling in the bispectrum. New things have happened since the original presentation of this webinar that warrants updating it. Main theme is still present, no matter which new modules are added to NeuroGuide, and the goal is to link patient symptoms to dysregulation of the patient's brain by forming hypotheses, and making sure you have good collection hygiene, visual editing of the EEG, making sure that there is no artifact and that one selects artifact-free sections of the EEG with at least 0.9 test-retest reliability, and preferably even higher. Then you do spectral analyses. This is in the light blue part here. And once you do those analyses, you evaluate what you have, and you then begin to link dysregulation and nodes and networks of the brain that are linked to the patient's symptoms. A good way to do this is to form hypotheses prior to recording EEG based on the patient's symptoms. So if the patient has a history of attention deficit disorders, and you'll look at the attention network. If it's anxiety, it will be the anxiety network. If it's memory problems, it will be the memory network, etc. Once you make that linkage and identify the dysregulated hubs that are related to the patient's symptoms, then you begin neurofeedback targeting those weak hubs and connections, reinforcing them towards greater stability and greater efficiency. And then, of course, you evaluate the efficacy of your treatment. You can do that by statistics with the brain itself. You have also symptom checklists or symptom charts of the severity of the symptoms, and you really want to see those go down. Now, today we're in the same arena of linking, but what we're going to be talking about is cross-frequency coupling, which is a very, very important topic that's new to many of you, but has been especially elaborated in the last 20 years as being one of the fundamental mechanisms by which the brain selects different modules across the vertical phylogeny of our brain. That is from the brainstem to the limbic system to the basic ganglia to the cortex. There's vertical organizations that are going on, as well as cortical synchronizations at different frequencies. And so we'll go into the details of that. What's important is, and I'll be emphasizing this throughout, still is location, 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 and not necessarily frequency, because every node and module and cluster of neurons actually communicates with other clusters of neurons at different frequencies. And they're completely capable of generating a broad spectrum of frequencies. And so location is what's important, and whether there's dysregulation expressed in a delta or a theta or alpha band in the hippocampus or the cingulate gyrus is much less important than the location itself. And this is emphasized in this paper that I published in a number of years back now. I guess it's 2015, on the mechanisms of neuron selection. And it's in the Psychiatry Journal, in which the cross-frequency coupling was emphasized. Now, what we're going to deal with today is two categories of cross-frequency coupling. One will be what's called the bispectral analyses, and the other is the cross-frequency analyses. Both of them are involved in the vertical connectivity from a reptilian mammalian to primate brain. The bispectral analyses are for the infra-low frequencies. It's a method to evaluate the relationship between the functional MRI bold, very slow changes in the synchrony of large masses of neurons, and the higher frequencies of that constitute the EEG, which is different than the functional MRI. But I'll show you how to use the bispectral analyses without having to use a DC amplifier, and to be able to look at very, very low frequencies and see how they relate to higher frequencies. And then, of course, just the straight cross-frequency coupling. And here you can see we're going to be talking about derivatives of the bispectrum, but we're also going to go into the cross-frequency power correlations, cross-frequency phase lock, cross-frequency phase shift, cross-frequency LORETA coherence, and cross-frequency phase-amplitude coupling. These are different ways of looking at the way low frequencies are coupled to higher frequencies and across both space and the frequency domain. So we'll go through each of these, but we also, at the end, I'm going to open up a NeuroGuide and show you how you can activate these modules inside of NeuroGuide. Now, one of the important discoveries recently is that the brain is actually not like a computer. Instead, there's matching and mismatching of expectations going on continually throughout the brain, and a continuous flow of information from sensory inputs to frontal expectations and behavioral selections and selection rules to meet the drives and needs of the individual. So it's an information flow dynamic of matching and mismatching, and novelty is absolutely fundamental to the creation of new synapses and changes of the size of synapses, and expectations of the future are very, very important. Now, there's an entire spectrum from the phylogenetic, really ancient organization of biological material, namely RNA, DNA, and the single cell. Then there's collections of cells, synapses in the brains which communicate through microcircuits, and then networks are formed, and then there's a behavioral expression of this dynamic of uncertainty reduction that's going on continually as people navigate in the world and meet their needs and drives. So this is an interesting way to see this this biological spectrum, and neurofeedback is primarily involved with the circuits and the networks, and quantitative EEG as well, where there's network oscillations, and we're going to go into a little detail of how neurons are selected to mediate a given function at a given moment of time. Since there's 100 billion neurons, there's subsets of these neurons that are synchronized for brief periods of time, released, and then another group is synchronized, and this is about for 200 millisecond or 250 millisecond frames of time in healthy brains. And you'll see that these lengths of time for synchrony vary depending upon symptoms and location. Now, how are neurons selected? Well, one thing that we've discovered is that there's a relationship between the probability of neuron firing and the local field potential. Now, the local field potential is the electrical potential that you can measure in the EEG frequency range with a microelectrode just outside a group of neurons. Now, the microelectrode is picking up the electrical field from a large number of neurons, but there turns out to be a correlation between the probability of discharge of the action potentials and the phase of the local field potential. If the phase of local field potential is excitatory, the neurons tend to burst. If they're shifted antiphase, then they will be suppressed. So we're going to look also at this spectrum from Varela et al. In 2001, which he was able to show this nesting of local field potentials where you can put electrodes inside neurons. You'll see that there's synaptic activity going on in the frequency range. You withdraw the neuron, the microelectrode, so it's just outside the neuron, and you pick up, that's in B, there is local field potentials. And the summation of the electrical field potentials, which can be seen here in IEEG with larger extracellular electrodes, is what the EEG is. And then from the scalp surface, you're looking at the summation of many more synaptic potentials represented by these local field potentials, which actually relate to the probability of discharge in large masses of neurons for brief periods of time. So how are neurons selected for brief periods of time? Well, the short answer is neurons are shifted in phase to the local field potential when they're selected, and they're suppressed by shifting them anti-phase. And this has been shown in many studies. This is a study by Hughes et al., 2004. This is reviewed in great detail in the Handbook of Quantitative EEG and EEG Biofeedback, but it's also available by a simple Google search or a search of the National Library of Medicine. So when neurons are shifted in phase, like over here, they tend to discharge when they're shifted antiphase of local field potential, then they are suppressed. So there's in phase, antiphase, and there's a shifting that happens for the neurons with respect to the local field potential by which they are then selected. There's another study showing that the probability of discharge varies as a function of the local field potential, both suppression and then also selection and activation when it's in phase. So there's many studies showing this. Now, what is phase difference and coherence? And I'm going to show you this. Many of you already know it, but it doesn't hurt to review it. Notice that if you use ICA or PCA reconstruction, you will have destroyed all of the phase differences, the time differences between groups of neurons in the brain, and you'll be left with information related to amplitude, but the network dynamics will have been completely distorted. That's why it's important not to use ICA reconstruction. ICA is okay to decompose like you would a linear decomposition of a complex set of numbers in order to reduce the measure set. It's very good for statistics, but the problem is it's used to do artifact rejection, alleged artifact rejection, but actually ICA reconstruction creates artifact. So the best way to do things is simply delete artifact, don't reconstruct something out of nothing. And then verify that you have EEG that's highly replicable with high test-retest reliability, and that it reflects the actual underlying physiology of the person's brain. Now, what is phase differences? Well, it's essentially, if you can imagine a wave, and you got two waves. Let's say the top one is O1, the bottom one, number 2 is P3. Then P3 is shifted, in this case, 90 degrees with respect to O1. So when O1 is at a peak, where phi1 equals 0 degrees, phi2 for the P3 electrode will be at 90 degrees because it's shifted by 90 degrees. So you can see how there's a shift of one with respect to the other. Now this is important because this is actually time. So if you have a 200, or say a delta, a 250 millisecond wave at 4 hertz, a 90 degree shift, or in the case of, let's say it's 180 degree shift, would be half of 250. So it'll be 125 milliseconds. So there's a relationship between time and the phase shift, and it's used in physics and in math, the phase shift, to be represented really as a circle. This is actually the complex domain of a circle and related to a wave. So that's why the degrees, and you can also talk about radians, and the ways to explain phase shifting. In addition, if you know the frequency, you can translate phase difference into a time difference. Now, coherence is a measure of the consistency of the phase differences. And everything's represented on a circle when you actually work with frequency and time domain relationships. In this case, the black dots are the phase differences. They're clustered, for example, right around this phase difference, which is about 60 degrees. And the length of this resultant vector is coherence. So when the phase differences are really clustered tightly, then the coherence is high. If the phase differences are spread all around this circle, okay, then this resultant vector will go to 0, or coherence will equal 0. So remember, coherence is a measure of the consistency of phase differences over time. Now, one of the interesting things is to measure the duration of phase shift and phase lock, and to dig down a little deeper, drill down into coherence in more detail to see what's going on for the selection of neurons at a given instant of time, and or the allocation of those resources to mediate a given function for a short period of time. And that's done by what's called a phase shift, followed by a phase lock. Overall, phase shift and phase lock are referred to as phase reset, and there's a very extensive literature on this topic in the scientific literature. It's also described in the Handbook of Quantitative EEG. an EEG biofeedback. This explains what phase shift and phase lock is. Again, we have in the upper left here, coherence represented by the consistency of phase differences. In this case, the resultant vector for this instant of time for time point 1, time point 2, time point 3, time point 4 is 45 degrees. And then suddenly there's a shift to 10 degrees for time point 5, time point 6, time point 7, time point 8. But coherence is about the same as even though there's been a phase shift. And what we're doing with coherence is summing it over time, so you tend not to see the actual phase shift because it's so short. But we can evaluate the phase shift by calculating the first derivative of the time series. So the first derivative is a measure of change. And so you can see here that time point 1, 2, 3, 4, and 4 have very little change. So the first derivative is close to 0. Now suddenly there's a shift. That means there's a change in the phase difference. So the first derivative is high. After the phase shift, then there's consistency, or there's very little change. So now the first derivative equals 0 again. So the first derivative is a good way, and the second derivative, I'll show that in a minute, good way to quantify the duration of phase shifting and the duration of phase locking. And this is how we do it. I'm not going to go into a lot of detail on this, but it's highly accurate. With the computer, you're able to calculate by calculating the second derivative, you can get the inflection point of a phase shift, and it allows you to measure the duration of that phase shift in milliseconds. And then at the termination of the phase shift, there's now phase lock, so you can measure the duration of phase lock. That's, again, where the first derivative is close to 0. In this case, we have a 5-degree threshold, so if it's 5 degrees, that's essentially close to 0. We played around with 3 degrees and 7 degrees. It's bouncing around a little too much to give it exactly at 0, but this is sufficient to quantify the duration of phase locking and phase shifting. 5-degree threshold is fairly common in the scientific literature. You can actually see phase shifting and phase locking in the records. In NeuroGuide, for example, you can plot the FP1 with respect to F3, FP1C3, FP1P3, FP1O1, and this is phase actually in degrees, and you can see there are phase differences between FP1 and C3, for example, here. Or there's FP1, F3, FP1C3, P3, and FP1O1 all have a phase shift at this instant of time. Well, if we calculate the first derivative of this trace up here, now we expose in great detail the duration of the phase changes. In this case, there's a phase shift followed by phase lock, and then there's another phase shift. But we can measure the duration of this phase shift and the duration of the phase lock, and we did this with a very large number of individuals from birth to 17 years of age. We published that. We published it as it's called self-organization, because the brain is continually going into approaching chaos, and then followed by stability, chaos, stability, chaos, stability. But the duration of these changes from high uncertainty to low uncertainty is going on continuously in the brain. But we asked the question, how is this related to anything meaningful to us? And we looked at traumatic brain injury, intelligence, autistic children, etc. And this is just an example of a paper that was published in Neural Image in 2008 on relating phase shift and phase lock to intelligence. And what we found was that in the upper row here is, and we found this with all of the short distances, 6 centimeter, that is like O1P3 or C3P3, adjacent electrodes in the 1020 system, that the longer the duration of phase shifting, the higher the IQ. So you can see phase shift 50 milliseconds to 70 or 60 milliseconds, and then the higher the IQ. Now, we reasoned in our paper that the phase shift is a shout out. It's saying who's available. It's mediated largely by the thalamus. And so at any given moment of time, neurons are not available because they're refractory or they're committed to other functions, or actually because they're energy deprived. If you use photobiomodulation, you can actually increase the total supply of neurons because you inject photons that interact with the mitochondria, creating more ATP. But in any case, that's the relationship between phase shift and intelligence. Because not every neuron is available out of the 100 billion, these shout-outs are asking which neurons are available. And once those neurons are identified, they become recruited and synchronized into a phase lock. And here you can see phase lock in the bottom row, and the shorter the phase lock duration, the higher the IQ. So a very efficient brain is somewhere around 200 milliseconds of phase lock, and not too long. If it's too long, then there's less flexibility and less resource available at the next instant of time. So the more efficient brain is one that recruits neurons, a large number, then has that synchronous group mediate a given function for a minimal amount of time. Now, we can also look at the fact that the brain is really made up of a series of clusters or hubs. These are groups of neurons that are connected in networks, and there's dynamic switching between the networks itself. And only EEG can measure the switching dynamics of coupling and uncoupling, which happens cross-frequency as well as within a given frequency domain. Now, these are Brodmann areas. I'm not going to go into details in this. Corby and Brodmann showed that there's cytoarchitectural uniqueness in these different parts of the cortex that mediate somewhat different functions. For example, the blue is the visual cortex. The architecture of the neurons here is very different than the parietal lobes, or different than the temporal lobes, or different than the frontal lobes. And these parts of the brain have different functions. And they reasoned in 1909 that in structure function, that if there are different structures, you would expect that there are different functions. In those days, they didn't really understand that very well. But in the decade of the brain, beginning in the 1990s, functional MRI, PET scans, and MEG and EEG all started to show that there were clusters in the brain, and they more or less correspond to the Brodmann areas as a good architectonic mapping that now is used worldwide. This is a summary of 30,000 functional MRI and PET studies across many different experiments. Basically, resting states will predict how somebody will perform in an active task. But you can see that the amount of blood flow that increases in different parts of the brain happens in a subset of clusters. And these are fairly large clusters. These are centimeters of synchronous blood flow changes in the brain. So not the whole brain. There's actually time differences and frequency differences, cross-frequency coupling between these hubs. And part of the thing that we're going to do today is to show you the tools you can use to identify some of the cross-frequency between the major network hubs that are related to function. And the efficiency of the communication within a frequency band and between frequency bands between these hubs is what gives rise to good, healthy functioning, and as well as dysregulation. You can see if it's deviant from normal, it's linked to symptoms. Now, a remarkable paper came out in 2008 by Hagman et al. Showing using diffusion tensor imaging, which is structural MRI, you can count all of the axons, and you got 100 billion, but computers are very, very powerful. Their methods are very powerful. And they found by counting the density, the spatial density of the terminal parts of the axons, that there are 6 major hubs. So you can reduce 100 billion neurons to 6 hubs. And you'll have to read the paper to see what they are. This is the cuneus and the visual cortex. And there's frontal and parietal, but particularly frontal hubs up here. And so you can read what these hubs, what these numbers, these nomenclatures mean. But just the fact that there are 6 huge hubs is pretty amazing. Now, they've been able to show that you can decompose these 6 hubs into smaller hubs, like the Brodmann areas, for example. But the Brodmann areas are really children of this family of one or more of these large hubs. Now, myself and colleagues asked ourselves, well, if there's clustering of hubs like this, then we hypothesized that one should be able to measure the differences in current density using LORETA and correlations between hubs using LORETA, because LORETA is a measure, it's low resolution electromagnetic tomography, is produced by synchronous synapses. So if there's clusters of synchronous synapses, 6 of them, we should be able to replicate that, because this is the diffusion tensor imaging is a structural imaging method. So it's showing you the structural connectivity. And so let's look at the functional connectivity, but we should be able to replicate the structural connectivity. And EEG is really good because not only does it structural connectivity, but it can show you functional and effective connectivity, which I'll be getting into in a minute. And sure enough, what we found is that we could replicate the Hagman et al. Study. We showed that hub 1 in LORETA is hub 1 in Hagman, or hub 3 in Hagman is a third cluster in LORETA, and we completely replicated it. Even in a single individual, we could see the existence of these structural hubs. So these are real and they're cross-validated by an independent imaging modality that has very great power in the time domain. And this is diffusion tensor imaging. It's a beautiful image. It shows you the U-shaped fibers up here that are connecting up different clusters. And then you get the long distance fasciculae, the frontal occipital fasciculus, the arcuate fasciculus, etc. So you got both short distance and long distance organization. And sure enough, the EEG replicates this fairly well. But it's important to note that there are now atlases this is by Deschamps and Catani, of the diffusion tensor imaging. And these are Brodmann, like for example, Brodmann 39 and Brodmann 45. This is frontal parietal, very, very important in intelligence, but they're able to show that these subgroups exist. And what's important is EEG has sufficient resolution to be able to look at centrovoxels, for example, of Brodmann 39 and Brodmann 45, and see how they're cross-frequency coupled, or coupled within a band. So one can look at using cross-frequency measures and effective connectivity, the magnitude and direction of information flow between these hubs. They can only be achieved by creating a marriage between structural imaging, in this case diffusion tensor imaging, and the EEG, which is more functional and effective connectivity. Now, I'm not going to go into the details of this. This is in Deschamps and Catani. They're able to show that there are certain core clusters that are related to different disorders, psychopathy, Alzheimer's, amnesias, autism, schizophrenia, depression, anxiety, attention, etc. So this is a very interesting book, atlas, to read for those of you that are interested in learning more. But it's because of the structural and functional connectivity convergences and cross-validations that it makes it easier for clinicians to link patient symptoms to dysregulation in the hubs, nodes, and connections between the hubs and your patient, and then reinforce towards greater stability and efficiency. There's just another view of the short and long distance types of connections. This is LORETA correlations, in which A is connections within the gray matter. You really can't see them. They're not myelinated axons. They're very short. B are the U-shaped fibers, and C are the fasciculae, the long distance. And what you can see this is frequency on the x-axis here from 0 to 40 hertz. This is distance from the superior temporal gyrus up to the occipital region, or back to the occipital region. And you can see bands of high correlation, low correlation, high correlation, low correlation, high correlation, low correlation, high correlation, low correlation. You can see at a distance is quite low correlation. The color bar tells you the magnitude of the correlation. Blue means it's quite low because the magnitude of the connections, the density of connections, decreases as a function of distance. But you can also see that here's the alpha frequency band that these these areas like the inferior temporal gyrus is communicating with other regions of the brain in the alpha band or in the gamma band or the beta band. So there's different frequencies of communication for each of these hubs. So these hubs are really generating a lot of frequencies, but particularly you can see the alpha, gamma, and theta gamma, theta beta correlations here, corresponding to the U-shaped fiber of the structural system as well as the long distance connections. And there's many examples of this in NeuroGuide. Anytime you run the source correlations, you will see this type of a pattern where the horizontal lines reflect those U-shaped fibers and the long distance cortical connections to the fascicula. And you can get some idea of the cross frequency coupling between different parts of the brain that will be operating at a variety of frequencies. We also evaluated phase shift and phase lock in the default network. And this is the default network that we looked at. And we measured phase shift and phase lock from the center frequencies of LORETA. And we found the phase shift duration is there. It's very precise. It's more quantal. These are quanta, and that there's very discrete periods of phase shifting for different combinations of the Hagman hubs. I mean, not Hagman hubs, but of the default network. So there's different specific periods of duration of phase locking and phase shifting, and in this case it's phase shift, between the Hagman hubs. And we went into great detail in this and fitted a mathematical model to it. You can see that there's an exponential relationship between the magnitude or the duration of shift and lock as a function of distance from any one, which appears to be related to the density of connections between these systems. I'm not going to go into more detail. You should read this. Now, what's important here is that there is a phylogenetic vertical communication between the brainstem, the reptilian brain, and the rest of the brain. And it's a two-way street, and in which the brainstem contains the neuromodulators. That's where the small nuclei in the brainstem will produce neuromodulators like serotonin and norepinephrine and dopamine and acetylcholine, etc. These are very, very important little hubs, and their axons then spread out to the rest of the brain to modify the number and size of synapses. But there's a continual vertical communication, and we also can, in this case, we're looking at the lizard brain. You can actually take the human brain and overlay it on the lizard brain, and you see that they're the same in the earlier phases of development. This is a primate brain. This is a lizard brain. Primate, lizard. Myself and Dr. E. Roy John wrote a book on functional neuroscience in which we go into great detail on this phylogenetic homology and how the brain evolved past the reptilians into the mammalian species, and then into primates and into people, human beings. And so there's this phylogenetic commonality. And if you have observed the behavior of reptiles, you'll see that they have preserved, or primates have preserved a lot of their behaviors, and certainly the early mammals. Now we know also that, just to iterate, the locus coeruleus, for example, is epinephrine. That's in the brainstem. That's an excitatory neuromodulator that is fundamental to wakefulness. And there's a big input into the hypothalamus that when hypothalamus becomes is inhibited, actually while we're awake, it becomes uninhibited as we get drowsy. But in any case, when we're doing neurofeedback, we are actually reinforcing dopamine that allows, that's whose job it is to change the number and size of synapses. And that's how neurofeedback and operant conditioning operates to move the brain towards greater states of stability and efficiency, because there's actual modification of the synapses and the connections. This is all from the Thatcher and John book, showing inhibitory and excitatory zones and through the cross-frequency coupling, actually. And we'll go into more detail on how we can measure the cross-frequency coupling. There's some interesting geometries involved in the limbic system, the hippocampus to the cingulate gyrus and the dentate gyrus, with the septum and the amygdala in between. The amygdala gets its primary input from the thalamus, and so does the occipital region, although the occipital region is actually the nucleus accumbens mediation to the medial frontal lobes through the ventral tegmentum. I'm not going to go into details on that, but this vertical phylogenetic of the nature of the brain is important to be aware of. And one of the interesting things is that the size of the limbic system is relatively invariant across phylogeny. And what's happened is there's been an expansion of the cortex with greater, it's a modular reorganization. Dolphins have a very large temporal lobe to do sound localization. Human beings have a very big frontal lobe to create planning and goal-directed behaviors. And we also can see that there's a nesting of frequencies in which the infra-slow, the really low frequencies are mediated largely by glia and blood, ATP. And then, of course, there's electrode metal ionic interchange with the scalp you have to be aware of. And it's also some coordination as the blood and energy supply comes in to the brain, then neurons are nourished and they start discharging. And so it's a slow frequency, in this case 0.001, but 0.01. When you get down to 0.001, that's often just the electrode potential. Then there's slow cortical potentials. They're largely reticular and reticular cortical in nature from 0.05 to 4 hertz, very important in the mediation of arousal and vigilance. And this is oscillating, and as we start to go to sleep and get drowsy, there'll be a shift in control as the higher level neocortex is no longer dominant. Now, the limbic loops are roughly 4 to 9 hertz. That's in the hippocampus and the supramarginal areas of the hypothalamus and also the cingulate gyrus. But theta is in all of the pyramidal neurons as well. It's just that there's pacemakers in the limbic system that tend to cause it to resonate and dominate in that frequency range. And then in the neocortex, you have local inhibitory circuits that oscillate roughly at 22 hertz and higher. This is the gamma band, really. It begins right, according to Hutchinson's work, right around 22 hertz, because there's a different circuitry that's involved. And where there's the higher the frequency, the smaller the domain of oscillatory neurons there are. Now, in between the limbic and the gamma, or the theta and the gamma, is this range from 10 to 20 hertz, 22 hertz, which is involved in neocortical limbic integration and also reticular cortical integration, in which the dynamic competition for available resources is taking place, where the local field potential is shifting neurons and selecting neurons to be in phase or anti-phase in a dynamic predator-prey ecological dynamic. There's both cooperation, competition, there's independence, and then there's predator-prey. So I don't have time to go into this in detail, but this is a very important dynamic that's going on at micro levels throughout the brain, but we luckily can quantify this. This is an interesting study of what happens when people are induced using propofol. The EEG, well, first of all, there's very little coupling between the gamma, this is frequency down here, and the delta. Delta is up here, gamma is down here. And it's blue because there's not a lot of coupling. But as you become induced and lose consciousness, here's a loss of consciousness right here, you see hypercoupling between the gamma and the delta, and the gamma and the theta. And that remains highly coupled until you stop the propofol, and now there's recovery of consciousness, and you decouple the neocortex from the limbic and brainstem. So essentially what's happening under under propofol is there's a massive simplification of the brain. That is, there's no differentiation between the neocortex and the reticular formation. They're all basically oscillating, hyper-coupled. And now they need to differentiate as you regain consciousness. There's differentiation and separation in the cross-frequency domain, and the greater the separation there is, and the greater the clarity of thought and functioning. You can see that after this period of time, you still have not recovered the pre-induction status of the brain, and so it takes time for the brain to recover and to decouple and differentiate cross frequency wise. Now there's the Bi-Spectra versus DC recording. The Bi-Spectrum is a way to do two power spectra. You do a power spectra, and I'll go into on one, and then you do the power spectra to get you the Bi-Spectrum. But and this is how you do it in NeuroGuide. You click View, Dynamic Bi-Spectrum. The Bi-Spectrum is actually looking at the spindle activity, and we're looking at neurons will tend to burst and then get quiet, burst and get quiet. And so what we're looking at is the bursting pattern. Now it turns out this is a fairly low frequency. This is 0.01 hertz. And we can measure that and quantify it using the Bi-Spectra, where we have the burst activity here, and then we can measure through joint time frequency analysis the ensemble or the average of these bursts. Now this is the inter-burst interval, and this is the intra-burst duration. So we can get information about these metrics. This is, for example, when you click View Dynamic Bi-Spectrum, you can get a view like this, where the frequency is 0.03 here, and it goes up to 5 hertz. This is 1 hertz. This is a log of frequency and a log of amplitude. And you can see there's this cutoff about a little less than 1 hertz, about 0.5 hertz, where now it's a 1 over F. See that shape? It's going down this way. It's flat up here because this is the glia bold energy delivery part of the infra-slow frequencies. And then there's a transition to the cortical and limbic and reticular neural dynamics across frequency coupling at about 0.1 hertz. This is the slow cortical potential range, and then this is 5 hertz up here. So you can see it's quite slow. This cutoff here is at 0.8 hertz. So that's the cross frequency. That's the Bi-Spectrum. You're looking at the burst duration and how often things are bursting, and what's the period of time that there's no bursting. You can also look at cross frequency power during wakefulness, drowsiness, and sleep. You can see that in sleep you have hypercoupling of the delta gamma with the theta, delta and theta with the gamma and beta. So this red here is a simplification of the brain, essentially. While you're awake, things are more differentiated. So we've changed this around a little bit. You can see this in NeuroGuide for click analysis, cross frequency, and you can measure phase reset, phase amplitude coupling, coherence, a number of cross frequency metrics, both in C-scores and raw scores. And so this is how you get the cross frequency. Click analysis, cross frequency, and then select the cross frequency you want to do. Now, another area I've not talked about is the phase amplitude coupling. This is an example of where, from Jensen et al., where this slow rhythm here is theta. Writing on top of that rhythm are beta bursts. And the number, in this case, this was a memory paradigm. It's called the Sternberg paradigm, where you present, let's say, 7 items in a display. There's a delay, then you present one of those items and ask the individual, does that match or mismatch what you just saw? If there's a, you could just have an item that is not in the information set. Now, and there's a limit to people's memories, but what they found is that as they increased the number of elements in the set, that then the number of bursts writing on top of this theta rhythm, this theta wave, would increase. So if you had 3 items, you'd have A, B, C. If you have 7 items, it'll go out to G. But it's coordinated by the rising phase of the local field potential of the theta itself. So the phase of the theta is determining the amplitude of the beta. Now, more recently, with microelectrode recordings, we can see that, in fact, layers 1 through 3 produces the gamma and the beta of the neocortex. There's 6 layers in the neocortex. Layers 5 and 6 is the output of the neocortex, and it is at lower frequency. That's delta, theta, and alpha 1. But there's a coupling between the theta, phase of the theta, and the bursting of layers 1, 2, and 3. In fact, it's been shown that there's a feed-forward high-frequency beta in the neocortex in layers 1 to 3 to layers 5 and 6. And that there's then a feedback from layers 5 and 6 to the limbic system, the basal ganglia, the reticular formation, thalamus, that then comes back up to layer 1. Now, layer 1 is also influenced by cortical connections, but they're bursting in these upper layers at the higher frequencies. It's the lower layers that are at the lower frequencies. And there are obviously loops within the neocortex, but this cross-frequency coupling is one of the foundations. It's phase-amplitude coupling, where the phase of the theta determines the local field potential shift in phase, producing bursting activity, and then suppressing of activity when it's shifted anti-phase. So that right here, you can see a group of neurons that have been shifted in phase, and then those here have been shifted anti-phase. Again, that's a selection process. It's dynamic. It allows neurons to be recruited, coupled across the vertical domain phylogenetically from reptilian to human, to primate and higher level primates and humans through these mechanisms. Again, what's good is that we can measure this, we can quantify it. This is an example of cross-frequency phase-amplitude coupling in the right parietal lobe. You got frequency on the x-axis for the theta rhythm, and then the amplitude, there's a frequency of the amplitude is shown on the y-axis. This, for example, is alpha-beta coupling. Here's delta gamma coupling. And we measure this in great detail. The phase is in the x-axis, or the color. So that this allows us to quantify, and we do this for all 725 subjects from birth to adulthood. We can calculate z-scores. You can do this for raw scores. So the important thing is that we do have the capacity to do this. This is at the surface. In the future, we're going to be doing this between Brahman areas. But it's a very important advancement, because we can translate this into biofeedback. We're going to create NF3, and you begin to evaluate the deviation from normal and the efficiency of the brain to cross-frequency couple, and then you reinforce, using neurofeedback, improved cross-frequency coupling and phase-amplitude coupling. And then we can also do cross-frequency phase shift and lock. I'm not going to go into detail on this, but it is available inside of NeuroGuide. There's a beat frequency. If you take 2 frequencies and mix them together, in this case it's 4 hertz and 7 hertz, then you're going to get a beat frequency that comes out anytime there's differences. But we mathematically take advantage of this because we can tell that the phase difference is constant. It's 210 degrees. It's a constant phase difference between the 4 and the 7. And what we do is we then calculate a derivative of the phase differences. And when the derivative is constant, then that means the second derivative is 0, and that means there's phase locking, cross-frequency phase locking. When the first derivative is 0, that means that there's no change in the derivative relationship to these two frequencies, 4 and 7. And so that's the duration of phase locking. Actually, the duration of phase locking is the period of time, the duration of time where the second derivative equals 0. And we measure this just like we do the other measures for phase shift and phase lock within a frequency band, but now we do it across frequency bands, and we can look at cross-frequency phase reset just like we would the other measures. And again, these are raw scores where the color here is correlation, and but we can put this into z-scores as well, and you can adjust these measures. This is a cross-frequency phase shift duration plot, a cross-frequency phase lock duration plot. You can save these, you can get temp to limited files, you can do statistics on them, etc. Now we also have cross-frequency coherence, which is important, as well as LORETA phase slope index. Now this is not cross-frequency here, but the cross-frequency coherence is clearly cross-frequency, and that's between the center voxels of the 88 Brodmann areas, or the hubs and nodes, so we can look at how frequency is coupled. This is important because you know that the brain is operating at these different frequencies, as I mentioned, and shifting neurons in phase to become part of a temporary ensemble is a fundamental vertical dynamic. Here's the subthalamic nucleus interacting with the cortex and the thalamus, with the amygdala being involved in its synchrony as well, and then direct coupling from the subthalamic nucleus to the cortex with cross-frequency coupling. So here's cross-frequency coherence in LORETA, where we order these by, here's a delta theta, theta alpha 1, theta alpha 2, theta beta 1, theta beta 2, theta beta 3, theta high beta, alpha 2, high beta. So you can see that the theta alpha is highly coupled in the left hemisphere, and the right hemisphere is even more coupled in this individual. Then of course we'll have the absolute magnitudes are in this color bar, but we'll have z-scores for this as well. In fact, these are z-scores now. And you can see there's a reduced coupling in the theta high beta and the alpha high beta and the theta beta 3 band. For Brodmann area 34, with respect to Brodmann area 4, there's very low coupling. There's 3 standard deviations below normal. And Brodmann area 34 is the temporal lobes, and it's also the medial part of the temporal lobes is the amygdala. So there's very low coupling in the hippocampus and the amygdala and the temporal lobes to Brodmann area 4, which is a motor cortical area, or it's a prefrontal cortex. And so in the future, we're going to have LORETA cross-frequency phase-amplitude coupling. We'll have LORETA cross-frequency effective connectivity, which is phase slope index. That's the magnitude and direction of information flow. So these are just a heads up on the future. We'll be working on these in the coming days and months. And we're also going to have neurofeedback at cross-frequency surface neurofeedback and LORETA cross-frequency neurofeedback, where we use all these cross-frequency metrics from the center voxel of the Brodmann areas, or in the case of the surface cross-frequency neurofeedback, from the electrodes placed on the scalp surface. And in that case, you don't have to have 19 electrodes. It can be a smaller set. You have to have at least 2. But when it's linked to the patient's symptoms, then you're in better shape. Now, let me just briefly show you NeuroGuide so you can see how we do this. Here's the Bi-Spectrum. I've already edited this data. I'm going to click View, Dynamic Bi-Spectrum. We'll look at absolute power. And so here's the 0.03 hertz here This is 0.1 hertz. You can see it's fairly flat up here because this is the glial interface and blood ATP interface. And now you get 1 over F happening at about 1 hertz. And these are the Z-scores. You can look at different ones. You can look at absolute phase. You can look at different frequencies. There's the theta. This is bursting activity, okay? And then we do the cross spectrum. We do the joint-time frequency analysis of the spectra itself, or the bursting activity of the envelope, and that's how you get the bispectrum. It's not the same as cross frequency, although you're able to select the frequency which you want to look at as a base frequency, and then you look at the frequencies of the bursting between, this is FP1 and the rest of the brain. Or if you want to look at C3, click C3. P4, click P4. Now, the cross frequency for LORETA is obtained by here going to cross frequency coherence, and you can get Z-scores, and we're going to be expanding that. But this is the cross frequency for LORETA at this point. All we have is coherence, but in the future we'll have the phase-amplitude coupling and cross frequency phase slope index, or effective connectivity. And you can go directly under analysis to power correlations. That's the one I showed for the wakefulness, drowsiness, and sleep, or coherence, cross-frequency coherence. This is from the surface. Phase-amplitude coupling, again, that's the surface. And phase reset, like I was showing before. You can control the color map settings here. I'm not going to bother doing this because it takes a little time, but anybody with the demo, this is the demo, can play around with it. So the cross-frequency is embedded in LORETA here, and there'll be more of those. And cross-frequency is at the surface. And then, of course, you have the dynamic bispectrum of, this is log spot, this is a linear plot. And so that is what's available in NeuroGuide now, and will be coming out in greater detail in the future with more advances and an implementation of this into neurofeedback, which will be unique. Nobody's done cross-frequency neurofeedback. Nobody yet has been able to analyze in as much detail the cross-frequency nature of the human brain, and to have normative data all the way from birth to 82 years of age, where you can see how this cross-frequency dynamic matures and how there are growth spurts and there are changes at different ages. And that's why a normative database is so important, because it age matches your patient to a reference group, since the brain maturates and changes from birth to adulthood. And so the full panoply of structural connectivity, effective connectivity, and functional connectivity can be evaluated. And just very briefly, imagine a sports stadium and a parking lot and a street connecting the parking lot to the sports stadium. That is structural connectivity. That's what you see with MRI, structural MRI. And if the MRI is the same whether you're alive or dead, and shortly after death. And so it's structural. Same with the street that connects the stadium to the parking lot. Whether human beings exist or not, that street will still be there. Now, if you watch the number of cars that fill up the parking lot, shortly thereafter, there'll be a lot of people in the stadium. And when people leave the stadium, then the cars in the parking lot disappear. That's functional connectivity. That's correlations. That's coherence. So it's functional. And a functional MRI does that, and so does obviously an EEG. The effective connectivity is a measure of the number of people that go from the parking lot to the stadium and the number of people that go from the stadium to the parking lot. So it's a measure of the magnitude and direction of information flow. So all three measures of connectivity, structural, functional, and effective, are available in NeuroGuide and also in the EEG.